Hey guys, welcome back to my YouTube channel Dr. R.kmedicos. In this video, we are going to discussing about the morphology and physiology of bacteria. So continue to watch my video at last and never skip this video. And if you are new at my YouTube channel, then please subscribe, like and share my YouTube channel and never forget to press the bell icon so that you will get notification of all video uploaded by me on my YouTube channel. Let's start this video without escaping the time. Before discussing the morphology and physiology of bacteria, firstly we will see some important points and that should be remembered in your mind. And first is the resolution limit of I is about 200 microns, right? And the bacteria are unicellular and do not show the true branching except the higher bacteria like Actinomycetes. Now we will discuss about the microscopy. In introduction, you should remember is that the Leeuwenhoek first observed the bacteria over the 300 year ago. And now the type of microscope, as you already know that there are mainly three types of microscope. First is optical or light microscope, dark field microscope and electron microscope. Now we will discuss about the strain preparation. Firstly, in introduction, you should remember is that the bacteria may be strained in living state in a special purpose. But the routine method of staining of the bacteria involve the dyeing and fixing a smear procedure that kills them, and the bacteria have an affinity for the basic dye due to the acidic nature of protoplasm, as you already know. Now we will discuss about the different method of staining. Firstly, we will see the simple staining. In the simple staining, this method, the dye such as methylene blue or the basic fuchsin are used for simple staining, and they provide the color contrast but impact the same color to the all bacteria. This is the disadvantage of the simple staining process, right? And in this way, by this process we cannot identify the different type of bacteria. Now the negative staining, in this method the bacteria are mixed with dye such as Indian ink or nigrosin. And what is the use of negative staining? This is particularly useful in the demonstration of bacterial capsule which do not take the simple stain. And now we will discuss about the advantage of negative staining method. And as you already know that very cylinder bacteria such as asparagines that are not demonstrable by the simple staining method can be viewed by the negative staining method. This is the advantage of negative staining method. Now we will talk about the third method, that is impregnation method. In this method, the cell and structure too thin to seen under the ordinary microscope may be visible only if they are thickened by impregnation of the silver on the surface. So this method is known as impregnation method. And what is the advantage of impregnation method? Such method, that is impregnation method, are used for the demonstration of asparagines and the bacterial flagella. This is the advantage of the impregnation method. This is all about the different method of staining of the bacteria. Now we will discuss about the different type of stain. And firstly, in introduction you should remember is that this stain impact the different color to the different bacteria. And two most widely used differential stain are gram stain and acid fast stain. Firstly, we'll discuss about the gram stain. It originally discovered by Christian Gram as a method of staining of bacteria, as you already know. Now we will discuss about the step of gram stain. And the first step of gram staining procedure is that the primary staining with a pararosaniline dye such as crystal violet, methylene blue violet or the gentian violet. And the second step after that the application of dilute solution of iodine. And after this process, the decolorization with the organic solvent such as ethanol, acetone or aniline is taken places. And finally, contrast staining with a dye of contrasting color such as carbon fuchsin, saffronine or neutral red. This is the step of gram staining procedure. And by fourth step, we can apply the gram stain on the bacteria and differentiate between different type of bacteria. Now we will discuss about the classification of bacteria on the basis of gram stain. On the basis of gram stain, as you already know that the bacteria are classified into two categories. First is gram-positive bacteria and the gram-negative bacteria. And what is gram-positive bacteria? Those bacteria that resist the decolorization and retain the primary stain appearing violet. And this type of bacteria is known as gram-positive bacteria. Well, the gram-negative bacteria, those bacteria that are decolorized by organic solvent and appearing red, then such bacteria is known as gram-negative bacteria. And the remaining part of this topic we will discuss in next lecture. I hope that this video will be knowledgeable for you. Thank you for watching my video at last, and please subscribe, like and share my YouTube channel and support me.